Medial refers to a body part being closer than another part to an imaginary midline dividing the body into equal right and left halves. The eyes are medial to the ears. Lateral refers to a body part being closer to the side of the body than another part with respect to an imaginary midline dividing the body into equal right and left halves. The ears are lateral to the eyes. Proximal refers to a body part that is closer to the point of attachment or closer to the trunk of the body than another part. The knee is proximal to the foot. Distal refers to a body part that is farther from the point of attachment or farther from the trunk of the body than another part. The foot is distal to the knee. Anterior refers to a body part that is more toward the front of the body than another part. The eyes are anterior to the brain. Posterior refers to a body part that is more toward the back of the body than another part. The brain is posterior to the eyes. Superior refers to a body part that is above another part. The trunk is superior to the legs. Inferior refers to a body part that is below another part. The feet are inferior to the legs. Superficial refers to a body part that is closer to the surface than another part. The skin is superficial to the skeleton. Deep refers to a body part that is more internal and farther from the surface than another part. The skeleton is deep to the skin. Ipsilateral refers to a body part that is on the same side as another part with respect to an imaginary midline dividing the body into equal right and left halves. The right arm and right leg are ipsilateral. Contralateral refers to a body part that is on the opposite side of another part with respect to an imaginary midline dividing the body into equal right and left halves. The right arm and left leg are contralateral. A sagittal plane divides the body into left and right portions. A coronal or frontal plane divides the body into anterior and posterior portions. A transverse or cross-sectional plane divides the body into superior and inferior portions.
An atom has a nucleus with positively charged protons and uncharged neutrons. Negatively charged electrons orbit the nucleus and are arranged in shells. The shell closest to the nucleus contains a maximum of 2 electrons. All others can hold 8. The number of electrons equals the number of protons, and inner electron shells must be filled before any electrons are added to shells farther from the nucleus. Atoms interact or bond with one another so as to fill their outer shells with the maximum number of electrons. Hydrogen's nucleus has only 1 proton, indicated by the +1. 1 electron orbits in a shell that would be filled with 2. 2 hydrogen atoms in close proximity can share their electrons, so that each has 2 some of the time. This sharing of electrons is a covalent bond. Here we see several molecules that are the result of covalent bonding. Another type of bond is the result of attraction between ions of opposite charge. Ions are atoms that have gained or lost electrons. Sodium, on the left, has shells with electrons numbering 2, 8, and 1. Chlorine, on the right, has 2, 8, and 7. Sodium would have a full outer shell if it lost the single electron in its third shell. Chlorine's would be full if it gained 1. An electron is transferred from sodium to chlorine. Sodium is now Na+, chlorine is Cl-. The electrical attraction between them is an ionic bond. DNA is composed of two strands of nucleotides wound around one another in a double helix. The outer longitudinal structure consists of alternating sugars and phosphate groups. Paired nitrogenous bases are in the interior of the molecule. If the molecule unwinds, its structure resembles a ladder with phosphates and sugars forming the sides. The rungs are the nitrogenous bases of nucleotides on opposite sides of the ladder. There are 4 different bases, and only certain pairs are possible in forming the rungs. Adenine is always opposite thymine, and guanine is always opposite cytosine. Catalysts are substances that speed chemical reactions without being used up in those reactions. In cells, catalysts are usually large globular proteins known as enzymes. There is a portion of an enzyme, its active site, that binds only to a particular molecule, its substrate. The substrate is a participant in the reaction catalyzed by the enzyme. The binding of substrate to enzyme causes a change in the shape of the enzyme, which, in turn, facilitates the forming or breaking of bonds by the substrate. The product of the reaction is released, and the enzyme is available once again. Because of the fit between substrate and active site, each enzyme is specific for a particular reaction. When a metabolic pathway has produced enough end product, it can be temporarily shut down by feedback inhibition. The end product binds to the first enzyme in the pathway. This inhibits the first reaction, and thus all subsequent reactions. The end product is no longer made. In some pathways, the end product binds to the enzyme's active site, blocking binding by the first substrate. In other pathways, the end product binds to an allosteric site, away from the active site. This causes a change in the enzyme's shape, once again blocking the active site. As the end product is used up by the cell, it is no longer available to bind with the enzyme, and the pathway resumes normal operation. All molecules are in constant motion unless at absolute zero. The motion is random, with molecules moving in straight lines until they encounter other molecules. The net result of this process is diffusion, the movement of a substance from an area where it is in high concentration to an area where it is in low concentration. When drops of dye are added to a beaker of water, the drops are areas of high concentration of dye molecules. They will eventually be evenly distributed among the water molecules, and although all the molecules continue to be in motion, no further change will be observed. This membrane separates pure water on the left from a solution of sucrose and water on the right. The membrane is semi-permeable. Water can cross it, but sucrose is too large for the membrane's pores. Water molecules will move from a solution with a lower concentration of solutes to one with a higher concentration of solutes. A solution with fewer solutes has more water molecules that are free to move about and follow their concentration gradient across a membrane. Thus, there will be net movement of water across this membrane from left to right. This diffusion of water across a membrane is called osmosis. Osmosis is of great importance in the water balance of all cells. A cell may need to have a substance cross the plasma membrane against its concentration gradient. This requires energy and is accomplished by active transport. The sodium-potassium pump is an example of this process. A large protein in the plasma membrane provides the doorway through which sodium and potassium ions can move. ATP is the energy source. Sodium inside the cell binds to the protein. The addition of a phosphate group from ATP changes the shape of the protein and the sodium is expelled. Now potassium binds to the protein. The phosphate is released, and as the protein returns to its former shape, potassium is moved into the cell. Sodium can once again bind to the protein, and the process repeats as long as there is a supply of ATP. This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis. DNA is composed of two strands of nucleotides wound around one another in a double helix. The outer longitudinal structure consists of alternating sugars and phosphate groups. Paired nitrogenous bases are in the interior of the molecule. If the molecule unwinds, its structure resembles a ladder with phosphates and sugars forming the sides. The rungs are the nitrogenous bases of nucleotides on opposite sides of the ladder. There are 4 different bases, and only certain pairs are possible in forming the rungs. Adenine is always opposite thymine, and guanine is always opposite cytosine. These complementary pairs of nitrogenous bases lie next to one another because their sugar-phosphate chains run in opposite directions. This anti-parallel orientation of the 2 nucleotide strands brings the bases into position for hydrogen bond formation. The sides of the ladder are twisted in a spiral fashion and make a complete turn every 10 base pairs. DNA molecules are extremely long, thin, and delicate. They are packaged so that they can fit in the nucleus and be protected from damage. DNA is attracted to clusters of histone proteins. A portion of the DNA wraps around a cluster twice. This occurs at regular intervals along the molecule. This shortens the DNA so it can fit within the nucleus. The chromosomes are still fairly long, and their movement during cell division is facilitated by condensation. Following replication, they loop and fold back on themselves, forming tight packages of DNA and histone. This shortens and thickens the chromosomes, so they are visible under a microscope at the beginning of prophase. The rule of base pairing helps explain how DNA is replicated prior to cell division. Enzymes unzip the DNA by breaking the hydrogen bonds between the base pairs. The unpaired bases are now free to bind with other nucleotides with the appropriate complementary bases. The enzyme primase begins the process by synthesizing short primers of RNA nucleotides complementary to the unpaired DNA. DNA polymerase now attaches DNA nucleotides to one end of the growing complementary strand of nucleotides. Replication proceeds continuously along one strand, called the leading strand, which is shown here on the left. The process occurs in separate short segments called Okazaki fragments, next to the other, or lagging strand on the right. This difference is due to the fact that DNA polymerase can only add new nucleotides to the 3' end of a nucleotide strand. A primer begins any new strand, including each Okazaki fragment. An enzyme replaces the RNA primer with DNA nucleotides. Then an enzyme called DNA ligase binds the fragments to one another. There are now 2 DNA molecules. Each consists of an original nucleotide strand next to a new complementary strand. The 2 molecules are identical to each other. Transcription is the process by which RNA is assembled from a DNA template. The 2 strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the two strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene. Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome. A single messenger RNA molecule can be translated a number of times and produce many molecules of polypeptide. As a ribosome moves along the messenger RNA, additional ribosomes will follow. The result is an RNA with several ribosomes along its length. These ribosomes have growing polypeptide chains of increasing length as they move from initiation to termination site. The group of ribosomes is called a polyribosome or polysome. A point mutation is a change in the nucleotide sequence of a gene. This can result from an error during replication that is not corrected by the usual repair mechanisms. Under normal circumstances, adenines and thymines should bind to one another, as should cytosines and guanines. In a base substitution mutation, a non-complementary nucleotide is incorporated into the new strand. The resulting altered triplet may code for a different amino acid, or even a stop codon. Here we see 4 codons and their corresponding amino acids. A change within the second triplet results in a change in the amino acid. None of the other amino acids are affected. Insertions and deletions result in frame shift mutations. The insertion of an extra nucleotide not only affects the triplet it is part of, but all subsequent triplets, thus drastically altering the protein product. The result is similar if a nucleotide is deleted. Mitosis is the process by which the contents of the eukaryotic nucleus are separated into 2 genetically identical packages. Chromosomes replicate prior to the beginning of mitosis. As mitosis begins, they condense and become visible under a light microscope. They appear as sister chromatids joined at the centromere. Mitosis is divided into 4 stages. During prophase, the nuclear envelope disintegrates and a spindle of microtubules forms. Centrioles may help organize the spindle, as in this animal cell. The chromosomes begin to move toward the midplane of the spindle. When they are on the midplane, with centromeres attached to spindle fibers, the second stage, metaphase, has been reached. Metaphase yields to anaphase as the centromeres separate, and the sister chromatids, now termed chromosomes, are pulled toward opposite poles of the spindle. During the final stage, telophase, a nuclear envelope forms around each set of chromosomes. The spindle disappears, and the chromosomes decondense. The result is 2 nuclei, each with an identical set of chromosomes. Meiosis is the process by which a diploid nucleus divides twice to produce 4 haploid nuclei. The divisions are called meiosis I and meiosis II. In the life cycles of diploid organisms, meiosis precedes sexual reproduction. Among animals, the products of meiosis are gametes, eggs or sperm. DNA is replicated prior to the start of meiosis. The identical sister chromatids are joined at the centromere, as in mitosis. Unlike in mitosis, homologous chromosomes pair with one another. These pairs intertwine during early prophase of the first meiotic division, and may exchange segments. This exchange is called crossing over. During prophase I, the nuclear envelope disappears, and the spindle forms. The homologous pairs lie side by side as they reach the midplane of the spindle and attach to spindle fibers in metaphase 1. Metaphase ends and anaphase 1 begins as the partners in each pair of homologous chromosomes separate as they are pulled toward opposite poles of the spindle. These chromosomes still consist of sister chromatids joined at their centromeres. During telophase 1, the spindle disappears. Nuclear membranes may reform, and the two nuclei, each containing a haploid set of chromosomes, are separated as cytokinesis divides the cytoplasm. Prophase 2 begins with the formation of a spindle, and the still duplicated chromosomes move toward its midplane. At metaphase 2, they are lined up and attached to spindle fibers. Anaphase 2 begins when centromeres separate and sister chromatids, now considered chromosomes, begin moving in opposite directions. During telophase 2, the nuclear membrane reforms, the spindle disappears, and cytokinesis divides the cytoplasm. The result is 4 haploid cells.